Are you there, class?
Yes, Doc.
Good afternoon. Good afternoon. Afternoon. Let's have chapter 40. We jump to chapter 40. How far are you in physio in membranes? Have you finished membranes and physio? Hello? You are already in membranes.
Done na po.
Drop na po, sir.
Done na po.
Doc.
Okay. So, let's start. Biomedical importance. Okay, as you can see there, they are highly dynamic fluid structure. Take note of the word fluid. So which means that the membrane is not rigid. No, the structures there, like some proteins, they can move around. Okay. Now, plasma membrane form around compartments around the cellular cytoplasm. Okay, so it compartmentalizes the cytoplasm from the interstitial fluid around it. Okay, it has selective permeabilities. Selective permeabilities, and they act as barriers, maintaining difference in composition between inside and outside of the cell. Okay. Now this is provided by your transporters and ion channels, which means molecules can pass through and in and out of the membrane through transporters and ion channels. So exchange materials, it also is responsible for exchanging materials with the extracellular environment by a process known as endocytosis and exocytosis. So when you say endocytosis, that means from the outside, it gets inside the cell. Exocytosis, from the inside of the cell, outside of the cell. Okay. Plasma membrane plays key roles in cell-to-cell interactions and also in transmembrane signaling. Membranes also form specialized compartments within the cell. Specialized compartments within the cell. Okay. Of course, you are familiar that the cell has what you call your organelles. And those organelles, like the mitochondrion, they also have membrane. Okay. Now, maintenance of a normal and extracellular environment is fundamental to life. Okay. Now it says there that life originated in in an aqueous environment. So we can say that the big environment, aqueous environment, would be the sea that surround us, the ocean. So this would run counter to some of our, religious belief that life started from aqueous environment. So coming from the sea, probably starting from one cell organism and becomes multicellular, and then at first free swimming, and then develop some appendages so they can crawl, and they come out of the sea. So their gills must have been changed into lungs, like the amphibians. And these amphibians, through evolution, started to walk on two legs. And so that's it. That's science. But religion is different. We started as is, created by our creator. Okay? So sometimes science and religion cannot, go hand in hand. Okay? So life, as we said, is originated in the aqueous environment. Enzyme reaction, cellular and subcellular processes in membranes are important in compartmentalizing the body water. Okay. The body's internal water is compartmentalized. Water makes up about 60%. No? Some textbooks say 70%. So I'll just say 60 to 70% of the lean body mass of the human body, and is distributed in two places: intracellular fluid and extracellular fluid. Okay. So intracellular, this is about 2/3 of the total fluids of the body, and provides a specialized environment for the cell to make or synthesize, store, and utilize energy, and to repair itself, to replicate, and to perform cell-specific functions. Now, what about the extracellular fluid? This compartment is about 1/3 of the total body and is distributed between the plasma and interstitial compartments. Extracellular fluid is responsible for delivery of nutrients. Nutrients like glucose, fatty acids, amino acids, and minerals, vitamins, various ions, and other molecules. Hormones especially, they are regulatory molecules which controls the functions of widely separated cells. And this extracellular fluid also removes waste material as a result of the metabolism or cellular metabolism, as well as carbon dioxide, and of course, toxic materials that has been detoxified and passes out through the kidneys. Okay. So the ionic composition of our cells, our body, no? Ionic distribution of intracellular and extracellular fluids differ greatly. As you notice in that table, the intracellular fluid is rich in potassium. You have seen this, okay? Rich in potassium, no? And magnesium. There you are. And these are potassium, magnesium are the ions, and the anion is your chloride, no? Chlorides. Oh, wait a minute. Phosphate, I'm sorry. Phosphate here. Okay, phosphate here. While the extracellular fluid, you have sodium, extracellular 140, and calcium there, compared to that intracellular is very small, and then chlorides as the anion. Well, others are distributed, but those are the most important, and they can define so many changes within the body. So they have to be at that particular amount, extracellularly and intracellularly. Okay. Membranes are complex structure composed of lipids, proteins, and carbohydrates. Right. Or what we call carbohydrate-containing molecules. Now, on the right side is a comparison of the mass ratio of protein to lipid. So if you start from the top, that's myelin, there's more of lipids than protein, because the ratio there is 0.23. So the ratio is protein to lipids. So there's less of lipids. So what's that again? Oh, more of lipids. Sorry. All right. If you look at mitochondrial inner membrane, it's 3.2. So more of protein than lipids. Okay. And then next to that, sarcoplasmic reticulum, that's 2, more of proteins than lipids. Okay. So that's the membrane protein content, which varies in different tissues. Okay. Major lipids in mammalian membranes are phospholipids, glycosphingolipids, and cholesterol. Phospholipids is made up of a molecule of glycerol, and then fatty acids, and then a phosphate, and then an alcohol base. Alcohol base, that means it has an OH. No? Okay. So we have here your glycerol. Have your glycerol. And this is your fatty acid occupying the number 1 carbon of your glycerol. And the number 2 carbon is another fatty acid. The number 3 carbon, you have an acid, but this is phosphoric acid. And attached to the phosphoric acid is an alcohol. Okay? Now, the fatty acid that usually attached to carbon 1 is saturated fatty acid. That means fatty acid with no double bonds. Now, number 2 carbon is usually, but not always, usually occupied by unsaturated fatty acids. This is the location of your unsaturated fatty acids. Now, of course, you're familiar with your unsaturated fatty acids, no? Omega-3, omega-6, omega-9 fatty acids. Okay. Now, this is glycerol. This is phosphoric acid. So, glycerol and phosphoric acid only is known as what? Glycerophosphate. Diba? There you are. Okay. Now, if it has fatty acid, you say phosphatidic acid. Okay. And if you have this, so it all depends now, what is this? Supposing this is an amino acid, serine. Serine has an OH, diba? No? So you can call this phosphatidyl serine. Now, if occupying here is choline, so you call it phosphatidyl choline. If it is ethanolamine, you say phosphatidyl ethanolamine. Okay? That's your phospholipid. Now, the other one, that is glycerophospholipid. The other one now is glycosphingolipids. So instead of a glycerol backbone, what you have is backbone known as sphingol. And attached to the sphingol, sugar. So ceramide is known as a fatty acid at sphingosine, as is a ceramide. Then it is decorated with galactose and glucose. So you have galactosyl or glucosyl-ceramide and gangliosides. Okay, all right, that belongs now to this group, Glycosphingolipids. And then you have your sterol, and the sterol is represented by your cholesterol. Oh, there you are. That's why you produce daily 2 grams of cholesterol every day, whether you like it or not. Your liver produces that because you will be secreting cells every day. And in order to form new cells, you need or you're required to have your membrane. And the membrane, one of the important components of the membrane is cholesterol. Okay? So there you are. You destroy cells, and then you are going to form new cells, and those new cells require membranes, and the membranes require cholesterol. Okay. Now, membrane lipids are amphipathic. Now, what do you mean by that? Okay, very good. You have hydrophilic and hydrophobic. Hydrophobic meaning does not like the
Water loving.
Heat.
water hater.
Does not like
Does not
Hate
like
water.
it hates the water.
hates water.
Non-polar, very good. Hydrophilic loves the water.
Water-loving virgin.
Polar,
Okay.
polar.
Okay, polar. All right. Another four now is amphipathic. Now, let's jump right away. So you will understand right away that one there now is a representation, no, of this one is the polar head. You remember?
No.
No? And this one are fatty acids. Okay? The polar head is glycerol. Diba? Polar head is glycerol plus the phosphate there. That's the phosphate. It is water soluble or polar head. Now, sensitive sa dani. Ikapal na gamay mo. All right, now, the polar head and the two fatty acids. Now, this fatty acid in this illustration is saturated. Saturated. Okay. This one with a kink, that is unsaturated because you have a double bond there. The type of double bond here is cis. The other type of double bond is trans. Now, if this is trans, this is treated as straight saturated. Yeah, looks like saturated. All right. Now this one, they are all saturated. The two are all saturated. Okay. Now the ideal would be at the number 2 carbon of your glycerol, you have to have an unsaturated, preferably your omega-3 or omega-6 fatty acids. Okay? All right. Now, so what's the use of detergents? Okay. Your coronavirus has an envelope. Now, because it is not a cell, it is just a protein. An envelope there is composed of a lipid material and proteins. So it's supposed to be a membrane, but it did not come up to become a membrane. So there is no cytoplasm in that virus. So it's just a protein molecule with lipids. So much so that that envelope can be destroyed by detergents or soapy materials. Okay? So that's how you can remove virus by detergents. Okay? Membrane lipid forms by layers. But first, let's look at the micelle. Here's the micelle. So again, you have there a glycerophospholipid, in which, again, this is the polar head, this is your glycerol, your phosphate, and material attached to the phosphate. We mentioned choline, ethanolamine, serine, there. And this is your fatty acids. So you look at why they form like this. Now, when they form like this, it means that they are put in the water. When they are put in the water, the polar heads are around the surface, supposedly surface, yes. Now this illustration, just to show, but there's another one here, so they are so close to each other, like that. You have another one there, another one here, another one here. Okay. The idea is to have a barrier. That is a hydrophilic molecules on the surface, because around here, this is aqueous solution. This is watery solution. Watery solution. And so you have so many of these there. And inside is a hydrophobic material. Since they do not like the water so much, they cling to each other. So no water molecule would be in between them. Now this illustration is just to show, like that. Ano man ini amnesia pirmi. Okay. So this is a micelle. That's how it is. You have only a single layer here. That's a single layer. No? Okay. Now, this is a bilayer. That's a bilayer. So again, this is your aqueous solution here, right? Aqueous solution around like that. And the heads of your phospholipids are exposed to the water, as shown here. The heads are there. And the tails are inside So you have here hydrophobic area, and this is hydrophilic. The other side is hydrophilic. So you have now a bilayer. Okay. Now, the membrane are good, are really not really a filter or what, but it permits ions, but selectively. No, its permeability is selective. And if you notice this is the permeability, no? Permeability coefficients from low permeability to very high permeability. No? Very high. Okay. So you can see here, sodium and potassium ions can pass through. Low permeability. And then chlorides. So ions have difficulty. No? Then glucose and amino acids, then urea or glycerol, and then indole and water. Okay. So they have high permeability coefficient on the side and very low in here. So ions that passes through needs to have, in order that you will have this sodium or potassium or chlorides to pass through, if it is needed, you need something to help them to get in faster. I will go into that. Huh? All right. Membrane proteins are associated with lipids, and they form bilayers. We have shown that. So membrane phospholipid acts as solvent for membrane proteins, creating an environment in which the latter can function. Different membranes have different proteins. Okay. So, as you see that the organelles inside the cell have membranes, and we say here that different membranes have different protein composition. So we can identify, like for example, when you want to do some work in breaking the cell, and because you want to go into the contents, because enzymes are compartmentalized. Like for example, in the cytoplasm only, you have your glycolytic enzymes. Okay. You also have the enzymes for the synthesis of fatty acids. While the inside of your mitochondrion, you have your Krebs cycle enzymes, and also the enzymes for oxidation of fatty acids inside the mitochondrion. Now, if you're going to break and you want to get the enzymes inside the mitochondrion, how would you be able that you are already at the point that the cell had broken, the cellular membrane had broken, but the mitochondrial membrane is still intact, and you do not know whether you have broken the mitochondrion. No? So we have what we call protein markers or molecules that are unique to that particular membrane. So if you can detect that, that means that you are already breaking that particular organelle with that particular marker. Get it? Okay, it says major functional molecules of membranes consist of enzymes, pumps, and transporters, channels, structural components, antigens, etc., etc. Okay, including receptors. Now, if that particular protein, which is located on that membrane that you are going to break, is already in solution, so that means that you have already reached the membrane that you have just broken. No? Okay. All right. Now these are markers of membranes of particular enzymes that are present. Okay, the plasma membrane or the membranes, no? You have nucleotidase, adenyl cyclase, sodium potassium ATPase. They are located on the plasma membrane. In the plasmic reticulum, you have glucose 6-phosphatase. Okay. You have the Golgi apparatus. You have these enzymes and inner mitochondrial membrane, ATP synthase. ATP synthase. So these are enzymes that you can determine that you are already on that particular membrane. Like for example, when you do the test and you have seen ATP synthase already, or ATP synthase, then you have broken the mitochondrial membrane because they are located at the inner mitochondrial membrane. Okay? Membranes are dynamic structures. That's what I mentioned earlier. It says fluid structure. No? So, if they are fluid, then the position of the proteins that are there on the surface or integral, which penetrates the membrane, no? It does not mean that they stay there, no, in that location. They move around. So there is what we call lateral diffusion. No? Lateral diffusion, so they move from side to side. Okay? And there we have what we call transverse movement across the membrane. That is flip-flop, transverse. No? Okay. Membranes are asymmetric structures. Okay. So the structure you find on the surface of the membranes is not the same as the structures inside or the inner surface, or the inside surface of the membrane. So from the outside, the structures there are not the same in the inside of the membranes. And the proteins, there are two kinds of proteins. We have what we call integral proteins and peripheral proteins. Now, as we said, the membrane is having also phospholipids So you can dissolve them by the use of detergents. Okay. Now this is your membrane. You have two kinds of protein. This is a protein which runs through the membrane, like this one. And they are termed as integral. Okay. This is also considered integral because it has penetrated, but it did not penetrate the other side, or the inside. Okay. Which is again integral. But the one on the surface, like this one, and this one, and this one here, are what we call peripheral proteins. They are attached only to the phospholipids. Now watch this. Proteins, there might be some carbohydrate there. Carbohydrate molecules, glucose or galactose. Okay. And this is your polar heads of your phospholipids. Polar heads. And these are the fatty acids. So it's a bilayer. Okay. The fatty acids and the polar heads of your phospholipid or glycosphingolipids. Okay. All right, artificial membranes can model membrane function. Okay. Now, some artificial membranes are now being used to deliver medicine inside the body. Okay, because they can pass through membranes, and if the medicine cannot pass through membranes, so they can be delivered by artificial membranes that can pass through your membranes. And then once inside the cell, then it breaks and releases the medicine or the drug. So these are being used now. They are termed liposomes. Still with a lipid bilayer, so they can pass through membranes. The advantage of the use of artificial membrane for biochemical study of membrane function are as follows. The lipid content of the membranes can be varied, allowing systematic examination of the effects of varying lipid composition on certain functions. Now, this is for research work. And you have purified membrane proteins or enzymes can be incorporated into these vesicles in order to assess what factors, example, specific lipids or ancillary proteins required to reconstitute their function. Also, you can deliver now what is needed by the cells that may have a problem of getting inside the cell. Now, the environment of these systems can be rigidly controlled and systematically varied. samples are ion concentrations and ligands, so you have a control. When liposomes are formed, they can be made to entrap certain compounds within the vesicles, such as drugs or a portion of a DNA, or let us say a gene, okay, and can be delivered to where it is needed. There is interest in using liposomes to distribute drugs to certain tissues. And if components, like antibodies or certain cell surface, could be incorporated into liposomes targeted to specific tissue or tumors, for therapeutic purposes. Okay. So, gene therapy. Now, although they are doing this already, but it is not commonly done. It's not commonly done. So there come a time that you can just pop up a pill which has liposomes in it, and the liposome has some genetic material, or deliver it directly to where it is needed. Okay. Now, this is the fluid mosaic model of a membrane structure is widely accepted. This is elucidated by Singer and Nicholson, 1972. Okay. And it is now widely used and accepted. The model is often likened to integral membrane protein as icebergs floating in the sea. So again, this tells you that the membrane is fluid in nature. Fluid in nature. So those integral proteins are just, they can move around on the membrane. Okay, so which means that this integral protein does not stay there forever. It can move either that way or that way, or lateral, or go this way, or go that way. And because the phospholipids also, they move around. Okay, and those, like this, for example, these are carbohydrate that is attached to a phospholipid. Then it is move around. Right. So it is dynamic. It is not stagnant. Okay. Lipid rafts. You know what is a raft, no? Okay. So they are just floating. Lipid rafts. Caveolae and tight junction are specialized features of your plasma membranes. So you have certain specialized structures in the membrane. Life rafts, one of them. The areas of the exoplasmic or outer leaflet of the fluid bilayer, enriched in cholesterol, sphingolipids, and certain proteins. They have been hypothesized to be involved in signal transduction and other processes. It is thought that clustering certain components of signaling system closely together may increase the efficiency of their function. Lipid wraps. Okay, so there you are. And this is your lipid bilayer. So look at this. There are structures that penetrates. So integral lipidated, meaning proteins with a lipid attached to it, and non-lipidated transmembrane signaling proteins. So they are, look, they are there, penetrating the membrane. This is the membrane. Okay. There, oh. All right. Cholesterol, sphingomyelin, and glycosphingolipid enriched membranes, microdomains here. That one there. Oh, someone is here. And we also have what? Lipidated peripheral membrane-bound actin binding signaling proteins. Okay. So we have here forms a multilipid rafts, red membrane sheeting. Okay, sheet membrane reading. That represent localized microdomains rich in indicated lipids and signaling proteins. Signaling proteins there, those that are colored blue, green, and yellow. Blue, green, and yellow. Okay. Lipid wraps are stabilized through interactions, either direct or indirect, with actin cytoskeleton, actin proteins. Very similar to the actin proteins in your muscles. Okay. Cavulae. What are cavulae? All right. This is how it looks like. So it has a depression. Now this is the membrane, huh? This is the membrane. So you have a dimple here. All right, there. And these are lined with cavulin protein. All right. Okay. Cavula is an invagination. So it invaginates like that. Invagination in the plasma membrane. The protein cavulin lines up here. Okay. There. There. Appears to play an important role in the formation of cavulae. And of course, as a